All right, active transport. So thus far, all the transport mechanisms that we described were passive, meaning that they don't require the use of energy. They also all are moving substances down their concentration gradient, following natural laws of physics to spread evenly throughout a given space. But some substances need to be moved against their concentration gradient, so we use energy for it and call that active transport, meaning we're actively using energy to move the substance. So one of the most common types of active transport involves proteins that serve as pumps. The use of ATP to pump the substance into or out of the cell against their concentration gradient moves them from a region of low concentration to high concentration generally. The sodium-potassium pump, or sodium-potassium ATPase, ACE being enzyme, it's a protein, and ATP, meaning it needs ATP to do it, is an example of an active transporter. The pump will constantly pump sodium ions out of the cell and pull potassium ions into the cell in order to maintain an electrical gradient across the membrane. And again, an electrical gradient is very similar to a concentration gradient, only it deals in charge. It's the difference of an electrical charge across a space. These pumps are very abundant in nerve cells, as they use electrical signaling to communicate all over the body. In this scenario, the electrical gradient exists between the outside of the cell and inside, with the inside being negatively charged and the outside being positively charged. The negative electrical gradient is maintained as 3 sodium ions are pumped out of the cell. So you can see ATP is needed in order to pump them out of the cell. And then ATP is used ATP is used to pump the sodium out of the cell, and then the 2 potassium ions come back into the cell also for each ATP use. So ATP is used to pump them out and in. And then this process is super important because it accounts for most of the ATP that is used in neurons. And then there's another case of active transport called secondary active transport. We won't get too into it, but this occurs when an active transporter elicits the action of a passive transporter. So in the case of the sodium-potassium pump that we're talking about, it pumps sodium ions out of the cell, maintaining a concentration gradient of more sodium ions outside the cell. A sodium ion channel that allows sodium ions to pass through down their concentration gradient freely, like we were talking about previously, will just be open and allow those sodium ions to come back in. And so they kind of work in concert together. They're not necessarily attached, but this is considered secondary active transport still because the use of one is eliciting the effect of the other. Okay, so now we can talk about endocytosis, which is still active transport. This endocytosis, endo meaning inside, and cytosis being cell. It's literally the cell importing substances into the cell. So the cell does this by using a chunk of the membrane called a vesicle, which is a membranous sac. So it's literally a chunk of the membrane that engulfs whatever material is coming into the cell. Phagocytosis brings large particles into the cell, and this is common among immune cells that eat up invading pathogens. And so you can think of that as cells eating solids. Pinocytosis or pinocytosis brings fluid containing dissolved substances into the cell. You can think of phagocytosis as eating substances and pinocytosis as drinking them. Receptor-mediated endocytosis is the third one, and it's stimulated when a ligand binds to a receptor in the region of the membrane associated with the receptor, endocytoses it, bringing the receptor and the bound ligand into the cell. This is important for red blood cells that contain iron-dependent hemoglobin. Iron is bound to the transferrin. It's a protein, and that iron transferrin has affinity for transferrin receptors on red blood cells, and those red blood cell transferrin receptors on the membrane bind to those transferrin iron molecules, and then the cell engulfs the entire receptor ligand. So you can kind of see that here. Think of the orange star as the iron, and then the transferrin together, and then there's a transferrin receptor on the edge of the red blood cell membrane, and once that makes contact, and maybe enough of the ligands make contact, the cell engulfs that and then uses that. Exocytosis is the opposite of endocytosis. It's the exportation of substances out of the cell. Substances are packed up into a membrane-bound vesicle in the cell and excreted out into the extracellular fluid. This is important in many aspects of physiology. We can see it in stomach and pancreatic cells that produce and secrete digestive enzymes to break down food material. You can kind of see that here. We can see little stained vesicles all in these pancreatic cells, and these are ready to be secreting digestive enzymes. We also see it in endocrine cells that secrete hormones that travel in the bloodstream to elicit effects in other places of the body. And we see it in immune cells that secrete histamine to aid in immune responses, such as when we get bit by a mosquito. All right.